We're going to have our first panel today, and it's going to be hydrology changes over time, and our moderator is Dorothy Cifuentes. She's a PhD from Davie, Florida. She's the supervisory hydrologist for the USGS Caribbean Florida Water Science Center. Dorothy is the Associate Director for Hydrologic Investigations at the U.S. Geological Survey Caribbean Florida Water Science Center. Since 2008, she has been supervising scientists and providing technical oversight on a wide range of hydrologic studies of significant relevance to water resources in the region. Dorothy shapes the science direction of the center in anticipation of future water resource science needs, while aligning center science needs with national priorities. With that, I'll turn it over to Dorothy.
Thank you, Stacy. The panel right now, it's actually going to be in two presentations, and the first presentation is going to have 3 different speakers. So I will introduce them first, and then when they're done, we'll move on to the second presentation. And I think unless we have a lot of time, we'll probably wait to take questions at the end, if that's okay with everybody. Okay, and I want to just say something about water. I know hydrology, no pun intended, can be a dry subject. It's, all physics and math. But water is extremely important. We know it's a powerful force, and sometimes we think of, tsunamis or major flooding, but it's a powerful force in many other ways. And Joe referred to some of the ways in which, in the name of what's conventionally called progress, we try to control water. We try to control it for supply. We try to drain it for either development purposes or for farming or to control disease. Water and the landscape and ecology are all inextricably connected. And it's a in the Everglades, it's a commonly expressed sentiment that you have to get the water right in order to get the environment restored properly. And so this sort of leads into our presentations for this section. And it's about the relationship between water and restoration, and restoring the lands and healing the lands from previous attempts to control water. It all comes back to the water. It all comes back to hydrology. And what's interesting about the 2 different presentations today is you're going to have a really interesting juxtaposition of different hydrologic environments. We're going to be talking about the hydrologic environment in South Florida, which is a subtropical climate. It also has very dry periods, but it's the gradient is also very important. It's very, very flat here, very low gradient. And so that sort of plays into maybe how we think about restoration, because water doesn't flow really fast, even though there's an abundance of it at certain times of year. And then the second presentation is in a much drier area out west in Arizona. And so you have a much, much drier climate, but you have a lot more topography. And I think we're going to learn a little bit about how those 2 things, the climatic conditions as well as the topography, play into how we think about restoration. So with that, I'm going to introduce our speakers for the first presentation. The first is Lisa Meday, who works for the Environmental Resource Management Department of the Seminole Tribe of South Florida. She's a Water Quality Program Environmental Protection Specialist, and she's been there since 2008, and she has a lot of regulatory compliance experience in public utilities before that. She earned her master's degree in geography from the University of Miami in Florida, but I will tell you that she has a PhD of knowledge, at least one PhD worth of knowledge and insight in her head. And she's a great resource for understanding the hydrology of South Florida. Another speaker in the group is Professor Esiobu from professor of microbiology and biotechnology at Florida Atlantic University, which is 1 county north of here. She's also the president and founder of Applied Biotech, Inc., USA. And she's an expert in molecular microbial ecology, and you're going to hear some really interesting things that her lab is doing in this first presentation. So she's trying to understand microbial interactions between a host and the environment. And she's got many trained graduate students in her research labs, and they're working on developing new and reliable ways of measuring and maintaining environmental health and sustainability. And the third speaker in this first group is Brian Scott Mercer. And he's director of microbiology at Florida Spectrum Environmental Services, and he's been doing that since 2016. He has a Bachelor of Science degree in biology from Florida Atlantic University, and he's currently working on his master's degree from Florida Atlantic University, also known as FAU, with a thesis pertaining to environmental microbiology and looking at different methodologies, traditional versus emerging and new methodologies for looking at environmental microbiology. So with that, I'm going to let the first presenters go. The title of the presentation is Seminole Native Area Water Quality Analysis and Interpretation.
Hi, thank you. Can you hear me okay? I'm Lisa, and I work for the Seminole tribe. Thank you very much. And we've got Brian, and we call her Dr. E. They'll be coming up next. I wanted to say thank you to Dorothy very much for helping us with this, being the moderator for the session. We have 25 minutes. I'm going to be setting the stage, and then Brian and Dr. E are going to be discussing the results. I wanted to take a minute to talk about how our program fits within the Seminole tribe. I'm Lisa at Water Quality. There's 2 technicians, Keith, I know is here, and Craig. Water Quality fits within the Environmental Resource Management Department, which has a side arm with the Water Commission that approves things, and we are fit within the HERO office, which is Heritage and Environmental Resource Office, and HERO merged environmental resources with tribal historic preservation. HERO goes under the executive director of operations, who then is responsible to tribal council. So the HERO mission is advancing tribal legacies. Their vision is to unify the environment and the heritage in service to the tribe with the values of self-determination, which is promoting and building capacity for tribal self-governance, sustainability, which is protecting long-term health of the tribe's cultural and environmental heritage, and transformational, fostering innovative and creative thinking to problem solving. HERO has 4 goals, or 5. Responsibly managing tribal resources, strengthening tribal autonomy and sovereignty, facilitating cooperative and effective relationships, and also providing and supporting educational opportunities. Background information for the Seminole tribe. The tribe has 5 reservations throughout the state of Florida with over 90,000 acres of land. It's Big Cypress, Brighton, Immokalee, Hollywood, and Tampa. The tribe also has trust lands in 3 other areas, which is Coconut Creek, Fort Pierce, and Lakeland. Our presentation is focusing on Big Cypress Reservation, which I might be saying BC, so if you hear me saying BC, it's Big Cypress Reservation. It's the tribe's largest reservation. It's about 52,000 acres. It's primarily wetland. There is the cattle and pasture is very important to Big Cypress, and also tribal enterprises such as the museum and Billy Swamp Safari. Big Cypress Reservation has a unique geographic location. I want to get my pointer here. Okay, to the north and northwest is agriculture, and to the south is Big Cypress National Preserve. To the east of Big Cypress Reservation are the water conservation areas. There's 3 of them. The largest is Water Conservation Area 3, which is at 925 square miles. The water conservation areas have mixed ownership between the state of Florida, the South Florida Water Management District, and some private ownership. Part of Water Conservation Area 3 is leased to the Miccosukee tribe. The purposes of the water conservation areas are to receive drainage water from upstream, to keep water away from the urban coast, and to deliver water to Everglades National Park. You might see this acronym, which is SFWMD, which is the South Florida Water Management District. And they have a relationship with the state of Florida. Normally, states are just the Department of Environmental Protection. The state of Florida does have a Department of Environmental Protection, DEP, and they're responsible for administering the water resources at the state level, and they exercise general supervisory authority over the state's 5 water management districts. The water management districts are responsible for the administration of water resources at the regional level. So the first of all 5 districts was the South Florida Water Management District. It was created in 1949 as the Central Southern Florida Flood Control District. It has a service area of about 31% of the total state land. It operates the service area primary water control system, and it manages water supplies year-round. The core mission areas for the South Florida Water Management District are water supply, water quality, flood protection and floodplain management, and natural systems. The South Florida Water Management District restores and protects the natural ecosystems, well, that's what they say, and the Everglades. And the Everglades restoration is the largest environmental restoration project in the nation's history. So above the water conservation areas, I can see why he stood. I'm going to stand this way. I'm sorry, but it's hard to stand. Above the water conservation areas are stormwater treatment areas, and the purpose of they are called STAs. The purpose of the STAs are to clean water coming from agricultural runoff from the north. And there's 6 STAs with a total of 56,500 acres. They're right here. Water flows from the areas into the STAs and then through the water conservation areas. Okay, am I too loud? Okay, sorry. STAs function by taking water that contains phosphorus and it flows into an STA. Then there's emergent aquatic vegetation like cattails and bulrush, and submerged aquatic vegetation such as algae and hydrilla. And between the plants uptaking phosphorus and the phosphorus settling into the sediment, water flows out of STAs with less phosphorus in it. There are some herbicides that are used to prevent invasive species from overcrowding in STAs. And this is an example of what an STA looks like. Starting yesterday, you got to see our rainfall. South Florida receives about 59 inches of rainfall a year. And that rainfall used to replenish the groundwater table and then flow south, generally over land, which formed the original South Florida landscape. This is a figure from 1900, before development, as Mr. Frank was saying. Water came from Lake Okeechobee, generally flowed this way, and went out in a couple different places. Currently, there's a system of canals that modifies the way water flows, and the general overland flow has been limited down. I like old maps, so this is characterizing Big Cypress Reservation in the area and data gaps. This map is from 1839, and Lake Okeechobee was known as Big Water, and the Everglades known was Grass Water, and there's a route here that they said that the Indians said that you could get through from one way to the other. But in here is an Indian camp, and this part says Big Cypress. This is from 1842. You can see it says Big Cypress. There's an Indian camp noted there. And now this is the Everglades, and this is called Lake Okeechobee. This is 1879, Lake Okeechobee, the Everglades, Cypress Swamps, and that would be the location of Big Cypress Reservation. It's kind of like the maps of Antarctica before they explored it. They didn't really know, they were just drawing lines. That's kind of the point of this. This is information from the Bureau of Land Management Land Survey in 1872. And these 2 properties are the current location of Big Cypress Reservation. And there's 2 notes written for all of it. One word is impracticable, and the other word is swamp. They never went in. 1930, this is another map from TIPO. It's showing the location of Seminole camps and the Everglades, Lake Okeechobee, and these main canals. And they've also added the OK Slough here. 1941, the location of Big Cypress doesn't fit neatly within a defined area. That's the nugget out of this. In general terms, it's in the Big Cypress region of South Florida, and the word impracticable is going to come back up. This is a current aerial of Big Cypress, and this is the agricultural area above it. Big Cypress Reservation, these are the water conservation areas, Lake Okeechobee, Big Cypress National Preserve, and the Everglades. The blue lines on this map show the primary channelization done by the state to move water around. So you can see how water's being redirected kind of away. The purple lines are secondary drainage systems which channel water into the blue. I'm going to go back. You can see that the Big Cypress has, this is the OK Slough here, and there's a shred of a remainder of a natural flow left, which does include, I think this needs new batteries, Big Cypress Reservation. Upstream of Big Cypress is, the yellow is sugarcane, red is row crops, orange are citrus groves, gray is improved pasture, light green is unimproved pasture, and where there's no fill, it's natural land use. The Big Cypress Reservation Native Area, which is the focus of the study that we did, is south. Oh, it works. It's south of this West Feeder Canal, and it's west of the North Feeder Canal. It's about 14,000 acres. The land is in natural condition. Water only comes into the native area from rainfall and internal overland flow. There's no off-site man-made drainage input. Microtopography, like what Dorothy was saying, from high to low, from this part of the reservation down to the lowest part of the reservation, it's about 4 feet in difference. So it's microtopography. It's also home to culturally important plant and animal species and cultural activities. This is the tribe's flux map, which is land use, the 2017 update. Dark green in the native area is mixed wetland hardwood. Mid green is cypress. Cranberry is pine flatwood. The blues are freshwater, marsh, and prairie. And this light blue, which is kind of like a green, yeah, right there, is wetland forested mix, and the purple is hydric pine flatwood. Our forestry program did a study in 2014 with the University of South Florida, and they examined aerials from 1940, 1953, going up to 2014, and made polygons of the vegetation types. One of the deliverables of that study was they coded the vegetation types being if they were hydric or non-hydric. Everything that's red is showing a change in vegetation type from hydric to non-hydric, meaning it's getting, the vegetation type is becoming more dry. The blue is non-hydric to hydric, and where there's nothing, it's clear that's no change from 1940 to 2014. I added this one because the 1940 aerial was a little bit pixelated. It wasn't so good. So in that same area, between 1940 and 1953, that's already a change, which is probably just an artifact of making the polygons from the aerials. However, you can still see between the 1953 to the 2014, there's a significant change. In terms of water quality regionally, the state of Florida, the South Florida Water Management District, controls water quality upstream of the tribes. The Seminole tribe and the Miccosukee tribe have documented concerns with water quality. The Seminole tribe upstream in the North Feeder Canal, which is right there, there's a landowner agreement with that upstream North Feeder owner, and that owner has a consent agreement with the state of Florida about water quality entering tribal lands. That same canal the North Feeder Canal travels down south all the way. The laser pointer is going to need new batteries. This is Miccosukee land. The canal ends south there, and it's the terminus, and so that water, which is high in nutrients, is changing the vegetation types for the Miccosukee, so you can see it from space. To address water quality and water supply concerns, the Army Corps of Engineers and the state of Florida, the South Florida Water Management District, as the local partner, started something called WERP in 2016, or planning for WERP. WERP stands for Western Everglades Restoration Program or project or planning. Both tribes are included, and the general idea for WERP is to take the water out of the canals, to improve the quality of the water, and then to return that water to the natural lands. There was a prior investigation, which is called the Western Everglades Task Force, that occurred a couple of years before that, which included the Corps, USGS, the district, USDA, EPA, National Park Service. The result of the task force was that there was too many data gaps to go forward. However, in 2016, the Corps and the district decided to move forward anyways. There's 4 different regions. This is called Alternative H in WERP, which is the newest alternative. The Region 1 is all agriculture. Region 2 is Big Cypress Reservation, essentially, and some of the upstream area. Region 3 is Big Cypress National Preserve and Everglades, and Region 4 is Everglades. The location of Big Cypress within WERP, the canal water is redirected to stormwater treatment areas, or STAs. So that North Feeder Canal up here, which, up there, that water is going to be rerouted up to the northern STA, which is 3,400 acres. It's going to follow that water flow path to the east and then go down into Water Conservation Area 3. The water in the west of the reservation here is going to be moved up to a new 4,000 acre STA, and it's going to travel, it'll be treated for phosphorus only in the STA. It'll travel through these natural buffer lands in that lighter green area, and then its first point of release is directly into the Big Cypress Reservation native area, and then on to the Big Cypress National Preserve. The Seminole tribe has water quality code. They have EPA treatment as a state, and there's tribal policy relating to water quality that says that it's the policy is to maintain water quality for the conservation of the habitat of culturally important fish and wildlife, the conservation of culturally important plant life, and to protect tribe members' rights to carry on traditional cultural practices. The Water Quality Code purpose is to protect tribal member health and welfare, aquatic life and wildlife within the reservations, wildlife and wild plants and wildlife, and the natural environment to carry on traditional cultural activities. The tribe also has narrative criteria. There's 2 different, it's free from and nutrient. The free from narrative criteria says that all reservation surface waters shall be free from substances attributable to wastewater discharges or other pollutant sources that cause injury to, are chronically toxic to, or produce adverse physiological responses in humans, plants, wildlife, fish, or other aquatic life, are unsuitable for aquatic life maintenance and propagation, and are unsuitable to support balanced indigenous populations of aquatic life. The narrative nutrient criteria also reflects back on natural populations and balance of aquatic flora and fauna. Oops. So within WERP, the pollutant of concern is phosphorus. It's the biggest concern for the state of Florida and the federal partners for Everglades protection. The STAs are designed to reduce phosphorus levels, but they don't affect nitrogen or potassium or herbicides or metals or salts or bacteria, and they also aren't designed to support pollution-sensitive, native, balanced aquatic flora and fauna. So in working with WERP, the Water Quality Program has a responsibility of looking to prevent harm to the Big Cypress native area. It's the only land the tribe has like that. It's been guarded for generations, It's home to culturally important flora and fauna species, and it's virtually unknown, getting back to that word, impracticable. The task then is to investigate the benefit of the additional water supply while preventing harm to the native area from any new surface water discharge from this STA. And the issue outstanding is the data gaps that was noted by the 2013-14 task force. So we planned an aquatic diversity study to gather some of that information. And our study questions are, what are the current native area water and soil chemistry conditions? What flora and fauna are those conditions supporting? Is that flora and fauna healthy and balanced? Is it pollution sensitive or pollution tolerant? Is it native or non-native? We picked sites representing all different vegetative communities and those that were shown to have changed or not changed, and it's all those dots as each one of the sites. And also, does what's there match what's being shown in WIRP planning, which is important. Our components to our study include macroinvertebrates, wetland condition indexing, habitat assessment, site water depth to ground, periphyton sampling, sediment, paleodiatom and carbon dating, fish diversity sampling, wildlife site utilization, vegetation documentation, an herbararium voucher collection, soil sediment, and surface water quality sampling, not just for phosphorus, but also for hydrocarbons, metals, pesticides, herbicides, general chemistry, dioxins, furans, PCBs, and finally, we did DNA sampling for the different types of bacteria, viruses, fungi, and parasites, and protozoa in the water in the native area. So this is just to take you on a tour of the native area, which you're going to be seeing tomorrow too, but this swamp buggy is called Big Bertha. It's Billy Swamp Safari's biggest one, and you can see that they are having trouble getting through. We had an experienced tribal member driver, and he was awesome. This was part trying to get to the sites that they were going to. So in terms of being impracticable, these are just some pictures of the different types of eco or communities that they saw while they were doing sampling at the 26 different places, to give you an idea of what it looked like. And some of the wildlife that they found was a crayfish, a really big frog, fish. Those are two different species of salamanders. One of them is called a two-toed amphuna, and the other one is a greater siren. And they found bones of those in in the tree islands in the archaeological digs. They haven't changed for 2 million years. They were the second most common food source. A lot of air plants, orchids. That's a snow fungus. That's some kind of red lichen. So we took samples for our study to look at the impact from the STA into the native area. Since the STA is not built yet, we looked at the canals. We didn't know what alternative was going to be chosen at the time, so we tried to define any of the canal water sources, and we also took 4 different samples in the native area. So BCSO4 is the North Feeder Canal as it comes into the reservation. BCSO6 is the West Feeder as it enters the reservation, and that's what it looks like at the North Feeder Canal looking upstream. This is the West Feeder Canal looking upstream. Confusion Corners towards the eastern side of the reservation. There's 3 different water inputs. One of them is BCSO1, which is G409. The source of the water is the state STA 56. That's the tribe's entitlement water supply. BCSO2 is the L3 pool as it enters, and BCSO3 is US Sugar. All of the BCS sites that I'm showing you are part of our regular fixed station water quality monitoring program. So we have data on this, but we've never done that intense DNA of the bacteria. So this is what BCSO1 looks like as it's coming in. This is what BCSO2 looks like as it's entering the reservation. And this is what BCSO3 looks like as it's It's actually looking upstream, but it's heading towards the reservation, into it. The native area sites, there's 4 sites. This area on the west, KB, KM, is for Kissimmee Billy Slough, which is an important historic, not only like ground elevation, but feature within the native area and further down. Also Cowbone Island, CBI in the middle, and the East Slough heading out. Oops, I went backwards. Oh, it's louder over here. This is from our QAP that we had to do for the study, and what it's showing is that for each one of the sites, their WERP gave it a land use code, and the tribe gave it a land use code. And so it's looking at those two. It's got the component of change. It has the soil code to define what it is, and then that other one I should have just deleted, but it's pop-up. What did it mean to us? So you can see for a CBI mid, WERP is calling it a cypress swamp. Our flux code has it as an inland pond and slough, and the Hendry County Soil Code is calling it a freshwater marsh. But it's all the same place. This is what it actually looks like. So that's about what does it look like, what is it, what's being planned, in planning, what does it say it is compared to what does it actually look like? That's what the site actually looks like. Heavy peat, deep peat, and both of the codes, mixed wetland hardwood and hardwood swamp, are kind of, they're relatively consistent. The Kissimmee Middle North Slough, it looks like this. Water, peat again. WERP is planning on, WERP thinks it's a medium hydroperiod marsh. Our flux code says it's a freshwater marsh. The change map is saying it's from grass to cypress, and the soil code is saying that it's a freshwater marsh. And that's what a freshwater marsh looks like. Cowbone Island mid looks like that. That does have some sand towards the bottom. And WERP is calling it a cypress swamp, and the tribe's calling it an inland pond and slough. No change. That's what a cypress swamp. East Slough out. Looks like this. Also peat. WERP's calling it a wet prairie. We call it cypress. We have it as no change, and the soil code is a freshwater marsh, which that's a wet prairie. So with that background, I'm going to let Brian and Dr. E come up to talk about what they did in helping us to try to figure out what the impacts for the water could be. Thank you so much. This doesn't really work.
Thank you.
Yep, that's to go forward, and that's to go backwards. Oh, thank you.
Thank you very much, Lisa. Good morning, everyone. So, in the past 2 decades or so, microbiomes have taken a major central role, central place in ecology. We're beginning to understand that the sustainability of forests, of rivers, in fact, of all of the biosphere, including us and animals, depends on the microbiome structure of the environment. And it was for that reason that Lisa brought our lab and Brian's lab into this analysis, the water quality analysis. So the question that we sought to answer, there were 2 major questions. We wanted to know what was in the canal waters and how that might impact the native area. And we also wanted to know if the content of the canal waters would have any health implications on the biosphere, on the biodiversity, the beautiful little squids and birds that she showed you. To answer those 2 questions, we used a cutting-edge approach in which we can sequence all of the DNA in the environment. And we have specific softwares that we use to align those sequences and to find out all of the microorganisms in the environment. So the first thing we did was to extract the full DNA to sequence. But then we thought that DNA could also be from dead animals or from dead microorganisms. So we did a more rigorous, stringent analysis where we extracted all of the RNA from the samples that we received from Lisa's lab. Only living cells can make RNA. So when we analyze the RNAs, we can tell you all viable organisms in the environment. The entire microbiome structure can be derived. So we did that, and we used computers to align the sequences that we generate, and then use that to tell you which organisms you have in the environment. The second question that we had to address was the implication. Is there any health implication associated with organisms we find in the canal waters? To be able to respond to that, we use quantitative PCR technique, in which case we would be able to advise that we have this X number of viruses or of bacteria, and that this might have implications or not. So here comes the results. Where do we point this? There. There. He gave me a new one. And yeah, he said to use this one. Okay, so yours is still working. We're going to then use...
The batteries for the laser pointer don't work.
Well, he said to use only this one. Okay, we're going to go back here. I'm sorry. Okay, back to the screen. So so we did the metagenomic sequencing of RNA, whole genome, and we did the qPCR. So this is the first result we obtained. Here you see the genome reads that we got from, I wonder which one will work. This one. Oh, this is still working.
Just for a second.
All right, that's good. So you can see on the right-hand side, you have samples from the reserve, the Big Cypress Reserve. From the native, from the, okay. We have the samples from the natural environment, the unimpacted, on the right-hand side, and on the left-hand side, you've got the genome reads from the canals. So all of these have got a lot of living microorganisms present. Brian will tell you some more. Go ahead.
Yeah, so this is kind of where the data begins. So I want to take 1 minute real quick to go over so you guys aren't kind of like bombarded with this, and so you kind of understand it more. So a genome read, when we refer to that for simplistic nature right now, we're going to say that it corresponds to one organism that we get. So you can see from the raw data that we have 6 million and down to 4 million is kind of what we're seeing. Okay, so this is showing the total bacteria that we get from this new technology. And again, on the right side, you have the natural samples, and on the left side, you have the canal samples. And the most interesting part about this, you see that there's more bacteria in the natural samples. And when people think about bacteria, they think bad and everything like that. That's just showing that the natural samples, the natural preserve is rich in microbial diversity, which is a good thing. In the canal samples, you see lower numbers, but again, this is just the overall of how many we got. All of these results that you're going to see is per 100 milliliters, just to kind of keep it on those that, you know, do the water quality testing with the 100 milliliter bottles. So this is 500,000, 400,000, and down to 100,000 that we could see with this technology.
In fact, vast majority of the genomes we generated from the natural environment could not be found. We couldn't find a match to those genomes in the biggest databases that we have, global databases we consulted. So there are many novel organisms in the natural environment that we were not able to resolve who they were, these organisms.
And then further data mining, we took the bacteria down and we looked for those possible pathogenic bacteria, possible risk organisms. And again, on the right, you have the natural canal to the left. And what you can see here is the ones that we found, you can clearly see in the canal, there's a lot higher values. I wish the pointer worked, but the little dots that you see here, are of another bacteria. It's just they were so high we put it on a different axis. But this is kind of showing the dots are actually E. coli. It's a strain of E. coli, which many people, hear E. coli, think it's something bad. But this is an environmental strain. The strain lives in the environment. It can come from different animals. It's not necessarily from humans at all. And you see it's in greater quantities in the natural samples, which it kind of makes sense. The biggest thing to point out here is one of the risk organisms we found was Microcystis aeruginosa. This is a cyanobacteria that we now know, there's a lot of data on it, that blooms like an algae would bloom. And this is a possible risk when you have it in the canal samples and you're thinking about putting it into somewhere where it's not. That doesn't necessarily mean that it will bloom. It could be outcompeted by other organisms, but it is a risk, and it's there in quite high numbers. Again, these are in 100 milliliters of sample, and you have the values there of 10,000, 6,000 organisms. And now, again, with this new data, this is all one test. So one test, we get all this information. Now we're looking at the viruses. And you want to take the virus?
So in general, you can see that the canal samples have many more viruses than the native areas. But you can also see that the native environment, or the samples from the native area, they have their own viruses. This is not uncommon. Viruses help to regulate the numbers of organisms in ecology. So they help to kill them when there are too many and to keep the numbers in balance. But what is surprising or a little worrisome here, this is not working. So you can see overall numbers of viruses, 33 from the BS2 and so on. But the high diversity of viruses present in the canal samples compared to the low numbers from the natural samples might pose some concern. Go ahead, they're giving us signs that we need to round up. Okay.
So the next thing we looked at, again, this is all one test, virulence factors. So I'll go real quick. Virulence factors are genes in bacteria that could pose potential risk to us. So they're genes that have processes in the bacteria, in the genome, that can pose risk. And you could clearly see, again, the natural samples on the right and the canal samples on the left, just the vast majority of them that are in the canal samples that are not found in the natural samples.
What is most worrisome, really, is not just the immediate assessment of impact, but the long-term possible impact in the environment. And that's where I want to draw your attention to the types of virulence genes we found. They were mostly born on plasmids and transposons. These are mobile elements. What this means is that over time, the microorganisms in the natural samples would be able to pick up those genes and express them. And you would have the biodiversity of bioresources in that environment perhaps impacted. But how long this would take to happen, we don't know.
So this is the second test that we did. So this is a separate test using qPCR for fecal biomarkers. So fecal biomarkers we use, one of them is really specific to human waste. It's the most specific, 99% confidence. And that's the one down here, W257. These other two up here are fecal biomarkers that have human implications, but also can be picked up from animal waste as well. So the main thing to point out here is these low numbers of results for the most specific to human waste. We don't really see any. Everything's under 10. When you usually get under 10, you call that kind of like insignificant.
So in conclusion, we found no significant human fecal pollution in the canal water samples as well as in the natural samples. That's a very good thing. We also found that the microbiological quality of the native area has a much more diverse, rich microbiome structure that's typical of an environment that's pristine, that's been preserved. I was quite impressed that we couldn't find a fingerprint of human pollution there. We are worried about the presence of Microcystis aeruginosa, which has the potential to bloom. While we're not saying that it would bloom, because we can't predict the environmental conditions that would allow its flourishing or diminishing, it's an organism to watch. That's an indication for continuous monitoring to watch what that organism might do to those beautiful waterways you saw. We don't want them blooming, becoming green and blue over time. The other important factor is the potential for virulence factors to mix in with the natural bacteria, the rich bacteria that you saw. We don't want them to pick up these virulence genes over time and express them, and that would impact the environment. So overall, we only sampled just 6 or 7 samples. We analyzed 6 or 7 samples. It's important to do many more samples. And the fact that we couldn't find many of those genomes in the databases means that we should take time to document perhaps potential organisms with rich bioresources that we don't have currently before we interfere with them by adding external samples. What is the next? And yeah, that was the, yeah.
I'm just going to wrap it up. I know we're a little bit of time. So the key points of this is, as they were both saying, in the native area, if the bacteria types are unknown in the world's largest databases, how can you understand what's present? And if without knowing what's present, because it's not in a database, how can you possibly monitor beneficial change through restoration or harmful change, meaning it hasn't been preserved? And then understanding what's different between the canal water and the native area water. Can we ensure that the water quality policy and free from standards are met? So in short, this meets our hero values of fostering innovative and creating thinking and promoting the long-term health of the cultural and environmental heritage. And it meets the hero goals of responsibly managing, strengthening sovereignty, facilitating collaborative and effective relationships, and supporting educational opportunities like this presentation. Thank you.